Mary Alex, and this is my TED Talk on what if we could rewrite our genetic future. Does anyone know the term impending doom? If not, impending doom refers to the feeling of dread. So, with that being said, imagine constantly living in a state of impending doom, knowing that one day the single gene inherited mutation that was written into your DNA from the beginning will take a toll on your body. For years, people and families have been affected by Huntington's disease and have had to live with that certain dread. Finally, history was made this year in gene editing. We have finally found a way to rewrite that genetic. The fairy godmother of genetics. CRISPR is rewriting genetic stories. CRISPR can modify DNA sequences with precision and efficiency. The CRISPR-Cas9 system can edit, delete, or deliver new genes. This technology uses a guide RNA to create a specific DNA sequence. This system forms two breaks in the target DNA, which triggers the DNA repair mechanisms. There are two major repair mechanisms, the non-homologous end joining and a homology-directed repair. The NHEJ repair places insertions or deletions, and HDR uses a donor template and then introduces an insertion. Currently, the main gene editing tool is CRISPR-Cas9. This tool continues to progress and hold promise for clinical use in the future for correcting inherited genetic mutations. CRISPR can help treat neurological disorders through gene editing. This system can also treat mutations through in utero, in vivo, and ex vivo editing. Currently, applications in clinical trials are used for cancer, immunotherapy, and genetic disorders. So, what is Huntington's disease? HD is a genetic neurological disorder that causes the progressive breakdown of nerve cells in the brain. It affects movement, cognition, and behavior. HD is an inherited condition caused by a mutation in the HTT gene on chromosome 4. This mutation leads to an abnormal repetition of the DNA sequence CAG, which causes the production of faulty form of the Huntington protein. Over time, this protein damages brain cells, leading to symptoms such as involuntary movements, difficulty with coordination, mood changes, and cognitive decline. It is an autosomal dominant, meaning a person only needs one copy of the mutated gene to develop the disorder. Symptoms typically appear in adulthood, but can occur earlier or later depending on the number of CAG repeats. Lastly, how can CRISPR help with HD? HD is caused by a mutation in the HTT gene where a DNA segment called the CAG is repeated too many times. This leads to a toxic form of the Huntington protein that damages brain cells, and CRISPR-Cas9 can target and cut this faulty gene sequence. By doing this, scientists can remove or shorten the extra CAG repeat, which reduces the harmful protein, turn off the mutated gene so it stops producing the toxic protein, and repair the mutation using the correct DNA template through homology-directed repair. Although CRISPR treatments for Huntington's are still in research and clinical testing, the goal is to prevent or slow down the disease before symptoms appear. 3, 2, 1, action. Here is a simple step-by-step diagram of how CRISPR works for HD. A guide RNA is designed so that it matches a specific sequence in the target DNA. The gRNA teams up with the Cas9 enzyme, or the scissors, and brings it to the exact spot in the DNA. Cas9 makes a cut or a double-stranded break in the DNA at the target location. Once the break happens, the cell's own DNA repair machinery is triggered. There are two main paths: the non-homologous end joining pathway, which is the broken ends are rejoined directly, often introducing small insertions or deletions that can disrupt the gene, or the homology-directed repair pathway, which is if a donor DNA template is provided, the cell can use it to repair the break and insert a correct sequence. By disrupting a harmful gene or correcting the mutation using these mechanisms, CRISPR can change how the cell behaves, like reduce the production of the disease-causing protein. A new chapter of healing. Until recently, Huntington's was considered incurable, but CRISPR technology offers a new hope. By precisely targeting and reducing the faulty HTT gene, researchers aim to slow or even stop neuron breakdown. This shift from an unchangeable genetic fate to a treatable condition represents a major step towards freedom and healing for patients and families affected by the Huntington's disease. The double-edged sword. Many ethical questions arise, especially when it comes to specific advancement. Should we change the human genome for future generations? Who gets access to these technologies? How do we balance innovation with moral responsibility? And ultimately, how do we ensure CRISPR is used to heal and not to enhance or divide humanity? One ethical concern surrounds the idea of the type of genetic editing. Somatic editing targets body cells like brain or blood cells to treat diseases in one person. The changes are not inherited. Germline editing, on the other hand, changes the DNA in reproductive cells like sperm cells, eggs, or embryos. This means the edited DNA is passed down to future generations. The major ethical concern is consent. Future generations cannot agree to these changes, yet they will have to live with the results. Philosopher Gingell raises questions about how we decide what counts as a disease versus a disability. For example, should we edit out only life-threatening mutations like Huntington's, or also traits like Fragile X syndrome or autism? This blurs ethical boundaries and can reinforce harmful social biases about what is considered normal or desirable. Gene editing therapies like CRISPR are expensive and highly specialized, which raises concerns about fair access. Wealthier individuals or countries might benefit first, widening the gap between those who can afford genetic treatments and those who cannot. This could create a form of genetic inequality where certain populations have better health or even enhanced abilities. The ethical issue in the situation is, should life-changing genetic therapies be a privilege or a right? CRISPR is powerful but still relatively new, and scientists don't fully understand the long-term consequences of editing human DNA. There's risks of off-target effects where CRISPR cuts the wrong part of the DNA, potentially causing new mutations or diseases. These edits could have unintended effects on gene interactions, leading to unpredictable outcomes over generations. Current in vivo delivery, like viral vectors, have limitations: immune responses and loading capacity. Non-viral delivery systems like lipid nanoparticles, polymer-based carriers are advancing, reducing off-target edits and improving fidelity via base editors, prime editors, which is a major goal. These are endless possibilities when it comes to this technology, but specifically for HD, technologies like CRISPR-Cas9 are rapidly advancing. It is becoming technically feasible to edit the human germline to prevent inherited diseases. For generations, Huntington's disease has written a story of loss, a story that families were forced to accept as unchangeable. But today, that story is starting to shift. CRISPR gives us the ability to reach into our own genetic code and rewrite what was once a sentence of fate. It's not science fiction anymore. It's science giving people a second chance at a future they thought they'd never have. Still, as we move forward, we have to remember that every edit comes with a choice that shapes not only our lives, but the lives of those who will come after us. The future of gene editing isn't just about rewriting our DNA, it's about redefining what it means to be human with responsibility, compassion, and hope. Thank you so much for listening.